This is the first ever study that shows mammals may be able to thrive in space. The International Space Station recently hosted mouse embryos successfully grown for the first time in the microgravity of space. Researchers had successfully grown mouse embryos aboard the International Space Station (ISS) for the first time. This represents the first ever study that shows mammals may be able to thrive in space. The University of Yamanashi and National Research Institute Riken said in a joint statement, adding that it is the world's first experiment that cultured early-stage mammalian embryos under complete microgravity of ISS. This new mouse embryo study is a big deal because it suggests humans can reproduce in space someday. Once the first human baby is born off the surface of our planet, we would become a true space-faring species. NASA's Artemis program aims to establish lunar camps. Those could ultimately pave the way for humans to stay in space much longer in the future, when the sci-fi vision of off-world reproduction may turn into reality. The experiment to grow the first mammalian embryos in space was led by Teruhiko Wakayama, a molecular biologist at the University of Yamanashi's Advanced Biotechnology Center, and a team from the Japan Aerospace Space Agency JAXA. The aim of the study was to determine whether a mammalian fetus can develop normally in the limited gravity or microgravity environment of space. To find the answer, frozen mouse embryos were blasted to the ISS aboard a SpaceX Falcon 9 rocket in August 2021. After arriving at the space station, the early-stage rodent embryos were thawed using a special instrument. Following this, astronauts cultured the embryos under microgravity for 4 days. The samples were then returned to Earth, where Wakayama and colleagues could study and compare them to mouse embryos grown in normal gravity here on terra firma. The team reported that embryos cultured under microgravity conditions developed into blastocysts, a cluster of dividing cells made by a fertilized egg with normal cell numbers. The researchers said that gravity had no significant effect on the blastocyst formation and initial differentiation of mammalian embryos. The team also found that, if allowed, the blastocysts would grow into mouse fetuses and placentas while showing no significant DNA alterations or changes in gene expression. The survival rate of the embryos grown on the ISS was lower, however, than those cultivated here on Earth. All this knowledge may eventually become very important to humans when we finally begin to think about sending people to the Moon and Mars for long stays or even permanent habitation. Thank you for watching. If you've enjoyed our videos so far, subscribe our channel for there's more to come.